Hi, hello. So I hope everybody would have seen the first video on our botany discussion where we have discussed what all things we must study in algae. So I am just beginning with one life cycle, Volvox. So let us see what all things we have to study in Volvox, and we'll see everything in detail. First of all, about Volvox, it is seen as a colony. So this is the colony. If you could see, this is the colony which is called synobium. Okay, and in synobium, the spheres, the small spheres that you are seeing on the surface of this spherical ball, that is like this. Actually, this is the individual cell. This sphere is actually this. Like, I hope you are feeling some familiarity with Chlamydomonas. Okay, yeah, it's looking like Chlamydomonas only. Actually, we'll see the parts of Volvox today, then what is the function of each of the parts, how it is looking like this, everything we'll see today. So, if I am zooming into this part, okay, imagine a spherical ball. These strands that you are seeing, these cells that you are seeing is in the surface of the ball. Within the hollow body of the sphere, there will be some substance called mucilage or gelatinous matrix. Imagine a rubber ball. In the hollow area of that rubber ball, you could see like gelatinous matrix or mucilages. Okay, and in that mucilage, you will see this area. Okay, this small darkened part you are seeing within that hollow sphere. That is the daughter cells of this colony. We'll study everything about it. Okay, let's discuss it one by one. So, though it is looking like sphere in the outer area, it is actually like this. See this image. So it's like Chlamydomonas, the individual cells, it is looking like Chlamydomonas. Just see this thing here. Okay, so it is an aggregation of Chlamydomonas like cells on the exterior, and there will be 500 to 60,000, that many number of cells, individual Volvox cells will be there. It will be connected by cytoplasmic strands. You could see this thing, this thing where I have focused. This is the cytoplasmic strand. Okay, so here these kind of cytoplasmic strands, how these cytoplasmic strands are created, that we'll study during reproduction of Volvox. So these kind of cytoplasmic strands makes it connected together, and it is acting like a colony where this is a spherical ball. All things are on the surface. Actually, even the individual spheres are these kind of individual Volvox cells. Okay, so everything what I am pointing here, everything is on the surface. Don't think that this is inside and this is outside. No, everything is like a on the surface of the sphere. Don't think that this area is within this area. No, everything is on the surface. Since it is a two-dimensional figure, we can display it only like this. Okay, let's move on. So Volvox is a genus of green algae containing around 20 species of freshwater algae. Thousands of cells together form colonies. Okay, you could see this. Okay, this is one colony, this is one colony, this is one colony, this is one colony, like that. So the greenish lighted area that you are seeing within is the daughter colonies. This is the outside colony, and this is the daughter colony. Once the outside colony bursts, this daughter colony will be released. So this is the realistic natural ecosystem where, like pond area, you can see the cluster of these Volvox colonies, and you see this is one colony, this is one colony, and this is the way it will be representing an algal bloom of Volvox in the pond area, etc. So Anton van Leeuwenhoek was the person who observed it and reported it first in 1700. So they live in freshwater habitats like ponds, etc. and all. Then, so this is the drawing that I have made. Okay, I apologize for the lack of neatness of drawing. Okay, this is the colony, and what is the outside circle that you are seeing? In all drawings, you will be seeing such an outside circle. This outside circle is actually this thing, this mucilaginous sheath, which is created around the each single cell of Volvox. This is the cytoplasmic sheath that is created, and this is the outside circle. Okay, then you can see the flagellated layer and all. We have already seen this image, and if I am zooming in to this area, you could see it is actually like this. So this is a zoomed out picture, so we are representing it like spheres. It is actually like this. Then, so it's again a spherical ball on the outside of the surface. You can see the flagellated individual Volvox cells. I want you to visualize this thing. That's why I am showing it again and again, the same thing. Then again, see, this is the outside circle. Then the individual daughter colonies are within. Once the outer exterior colony bursts, these individual colonies will be released. So this is the microscopic image. Then, so this slide I will be explaining during the reproduction. You must be curious how these inside ball or daughter colonies are created. If I am explaining it very briefly now, you could see this. Imagine this is a rubber ball. It is actually having a posterior end or a front end and an anterior end or a front end and a posterior end on the, one second, sorry for that, yeah, and a posterior end. This is the posterior end, then the lower side, and this is the anterior end, the front side. Okay, and in the front side the cells are somatic. That means it is not having the capacity to divide or reproduce. It is just for coordinating the motion or movement of the rolling motion of the Volvox. But the cells at the bottom side or the posterior side, it will be enlarging like this. Okay, the enlarged Volvox cell at the posterior end or the lower side end is known as gonidium. So within this gonidium, the Volvox will be dividing. See, you could see this. One cell divided into 2, then it became 8, then it became 16, then it became 32, like that. It will be dividing and it will be forming a colony like this. Then I'll explain during the reproduction session. I'll explain why it is showing an inversion like this. Then finally it will be turning like this. Then this loop will be closed, and finally this outer body will be burst and this colony will be released. Okay, now you can see that this colony is within the parent colony. This is the parent colony. Now this colony is within the parent colony. So that's how this kind of round spherical bodies will be created within the spherical body. We'll see that during the reproduction session. Then, so this is the individual Volvox cell. You can see this. Now, what are its parts? It is having an outer cell wall. Then it is having cytoplasmic strands. This is one cytoplasmic strand. This is one cytoplasmic strand to connect between individual cells. I have just zoomed into one cell to explain you the parts. That's why I have drawn it like this. You could see two flagellas, and these are called whiplash flagella. Then below the whiplash flagella, you can see contractile vacuoles, which will be coordinating the movement of the flagella and the entire Volvox cell. Then you can see a nucleus here. Then this entire cup-shaped structure is called chloroplast. Okay, which will be doing the photosynthetic function, the fixation of carbon dioxide and all. And during photosynthesis, carbohydrates or starch will be created, and it will be stored in this body within the chloroplast, that is called pyrenoid. Okay, then another structure part of this Volvox body is the eye spot. Eye spot, which will be sensing the light and which will be helping in the overall function of the Volvox cell. It will be also helping in photosynthesis as it will be receiving sunlight and all. So this is the structure of the individual Volvox cell. Now about the systematic position. So our answer, like till here I have just explained to you. Before moving to the answer part, I just wanted you to visualize things. Now we'll be going to the structure of the answer. The first account that you have to write during the note or answer writing in about a Volvox or any other genus is the systematic position. So the domain is, say, eukaryote, it is having nucleus. Then kingdom Plantae, phylum Chlorophyta, class Chlorophyceae, order Chlamydomonadeles. Okay, they are coming under the same order, Chlamydomonadeles. Okay, then there will be family Volvocaceae, then genus Volvox. So under genus will be having multiple species: Volvox aureus, Volvox lobator, okay, Volvox carpteri, Volvox barberi, etc. You can study just two examples of species, Volvox aureus or Volvox lobator. Volvox aureus is very common. It is causing harmful algal bloom in the ponds and all. So this is a systematic position. This you have to write for every genus that you are going to write a life cycle about. Then salient features: a single colony of Volvox looks like a ball of 0.5 mm in diameter. This is an extremely small like synovial colony, which is having, actually it is microscopic, but the colony we can see with our naked eyes, but the individual cell is like completely microscopic. So even then the entire synovial colony is just having approximately 0.5 mm in diameter. The plant body of Volvox is a hollow sphere called cenobium. Thousands, close to 60,000 cells are arranged in the periphery of the sphere. So that's it. Then the cells of cenobium are of two types: germ cells and flagellated somatic cells. I have already told you, at the anterior end you will be having like somatic cells which are flagellated. At the posterior end you will be having this kind of cell. One second. Yeah, this kind of cell. See, this is the synovial germ cell which is not having flagella. Okay, this is called gonidium, and it will be dividing to create new synovial colonies that we have discussed. So yeah, so there are two types of cell. On the anterior side you will be seeing the flagellated somatic cells which is not having any reproductive function. Then at the bottom side, or at the lower side, okay, or at the this side, posterior side, you will be having this kind of gonidal type of cells which will be having reproductive function. Then the extracellular matrix of a Volvox synovium. Okay, extracellular matrix means these are the individual cells. This is one cell, this is one cell, this is one cell. So what is between this thing? Okay, what is the inside content of the synovium? I told that it is a hollow sphere, so that hollowness will be filled with a extracellular matrix, because this is outside the individual cell, and that particular substance that is filled in the cavity or hollowness is known as mucilage. Okay, it's a gelatinous or like mucilage like substance which is secreted by the cells. The mucilage provides structural support to the colony and helps hold the individual cells together. Individual cells are spherical in shape. Okay, then they have cup-shaped chloroplast. Okay, they have cup-shaped chloroplast. Okay, they have cup-shaped chloroplast. And you have to take care of one thing. I told that individual cells are spherical in shape. This is not completely true for all the cells. The cells of the gonidium, you can see these are actually spherical in shape. But the cells, the flagellated somatic cells, these are like something like a pear shape or chlamydomonas like shape. These are the flagellated somatic cells. This is not spherical. This is a chlamydomonas or pear like shape. Then they are having a cup-shaped chloroplast. It is containing pyrenoids. It is having a nucleus, vacuole, and these are contractile vacuoles, and an eye spot. Okay, so two equal-sized flagella are present, that we have discussed already. Then each cell performs all the metabolic functions. Okay, since this is a green alga, it will be performing photosynthesis, respiration, excretion, everything. Cytoplasmic strands are formed during cell division. You see, this is divided together, so that's why a connection is established at this stage. This is the connection that you are seeing here. Okay, when it is mature. Fine. Then another thing interesting to note is the polarity that is existing in the synovium. What is the polarity? It is simple. The anterior end, this end is having elongated or pear-shaped chlamydomonas type cells, which is having the function in the locomotion or the movement of the like entire colony. While at the posterior side, you can see some cells which are having function for reproduction. So since it is having two different ends, it is having a polarity. So this is the posterior end where the cells are uniform in size. Okay, and it will be something like larger cells also. Then here at the posterior side, this is the lowermost side. This lowermost side, posterior side, you will be having gonadial cells. This is actually in the normal times smaller in size than the anterior somatic cell, but during reproduction its size will be increased to have the division capability and all that we will see during the asexual and sexual reproduction. Then eye spot. Eye spot is used for light reception. Cells with larger eye spot, this is the eye spot. Cells with larger eye spot are grouped together and they facilitate the phototaxics or the light recepted movement. Then Volvox significantly contribute to the production of oxygen. Since it's a green algae, it is performing photosynthesis and it will be releasing oxygen, and it is also acting as a food. This Volvox is acting as a food for many aquatic organisms. And the negative aspect of Volvox is that it will be, some species like Volvox aureus will be multiplying rapidly, and it is resulting in the harmful algal bloom. So you can see the greenish harmful algal bloom. So this is all about Volvox. So what we have studied is the entire answer to write for every kind of questions that can be asked related to the structure of Volvox. In the upcoming sessions, we will be studying about the asexual and sexual reproduction, and that will complete the Volvox. Hope you enjoyed the video. Thank you.